This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 283, recorded on May 2nd, 2014. Hey everybody, I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today from southeastern Michigan, Kathy Spindler.
Hi, everybody. You got it right. I'm in southeast Michigan, yeah.
I changed it. Well, I wrote it in the show notes because I want to be consistent with everyone. Mm-hmm. How's the weather out there?
Oh, we've got 55 degrees Fahrenheit, 12 degrees Celsius, and dark clouds and sun. It's kind of a mixture right now.
Oh, well, here it's really cloudy, but it is 20 degrees C, which is 68 F. Mm-hmm. It's one of the few centigrades that I know exactly, right? 0, 20, and 37. I didn't get to 37 very often. And partly cloudy. Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
How have you been, Alan?
Doing okay.
Yeah?
Yeah, things are good.
Do you have cloudy skies?
Oh, intermittently cloudy. It's some white puffy cumulus. It's probably going to rain a little more at some point, but right now it's nice and sunny, 18°C, gentle breeze.
We had such a deluge Wednesday here, did you?
Oh yeah, we got drenched.
Man, everything was flooded.
Yeah.
It's real. I drove to Princeton yesterday and all the roads were flooded and everything was overflowing. We have a guest today joining us from Frederick, Maryland. He is at he's at the NIH-NIAID. He's the lead virologist for the Integrated Research Facility at Fort Detrick. And he's got some other titles, which we will let him explain. Jens Kuhn, welcome to TWiV.
Hey, everybody.
How are you doing?
Oh, I'm fine.
Yeah,
the weather is not that nice, as far as I can see. Everything is like cloudy. And opposite to you, I know only centigrades and don't do the Fahrenheit spell, so it's about 19°C.
19°C, yeah. And so you're in Frederick, Maryland, right?
Correct.
So I got your title from a website. So that lead virologist part was right, correct?
Yes, that's correct, but I have to put the little disclaimer in there. So I work at NIH, not for NIH. Actually, for NIH, not at NIH. So I work for a contracting company, for Tunnel Consulting. And so I basically have two titles. I have a title for Tunnel and a title for the NIH facility, and lead virologist is my NIH title.
And so you work for Tunnel, but you're physically at Fort Detrick, right?
Correct.
Okay.
Most of the people in our facility are contractors.
Really?
Yes.
Why is that?
Oh, I think it's just the way how NIH tries to handle different facilities where a lot of different expertise is needed. Okay. So they out-consult and find consulting companies that, like Charles River for animal work So there's a lot of different contracting companies involved with this.
So you're not a government employee then, right?
No, we have only very few, about 5 or so.
That's probably good, right?
Well, it has advantages and disadvantages, right? I mean, the disadvantage, of course, is that you have several bosses. Yeah. And whenever you have several bosses, you get sometimes conflicting messages.
Yes.
The good news about it, of course, is there's overall more freedom and more flexibility. So, yeah, I mean, it's good.
So, Jens, tell us your life history. Where were you born and raised?
I was born in Germany, in Erlangen, in Bavaria.
Great beer.
That's right. Great beer and great bread and some good chocolate, yes. And then, I was pretty much my entire youth I spent in Nuremberg, kindergarten, elementary school, high school, and so on. And then when I discovered my love for microbes, I moved to Berlin and did all my graduate studies in Berlin.
How did you discover a love for microbes?
Serendipity, I guess. So I was a really bad high school student. So my grades were really terrible. And so I really didn't see it in my future that I would ever go to university and study or anything. But when I grew up, there was still compulsory military service in Germany. Mm-hmm. And if you didn't want to do military service, you had to do civic service instead. And so I chose civic service and ended up in a hospital in Nuremberg in typical clinical setting. and I was assigned to a microbiome lab. And my main job at that time was, well, what you can give an 18-year-old or 19-year-old to do in a microbiome lab. So I collected samples from different clinics, stool samples and urine samples, and drove them with a bicycle from one building to another. And so it was not very exciting. But I was lucky, and the technicians in the lab were not so much. There was some kind of a common cold thing going around, and a lot of the techs got sick, and so they were very short on staff. And so one of the attendings over there asked me whether I would like to help out in the lab, and not knowing what that meant, I said sure. So I ended up in the feces lab, and so I started spreading feces on agar plates and looking for salmonellas and campylobacters and all of these other little buggies. And it turned out that this was one of the few things that I'm actually not bad at. So feces spreading, that's right. so in the end, and I really enjoyed it, and so I liked the detective work in the end. So here's a colony, and what is this? And so, of course, at that time, we didn't have any PCRs or anything in that lab. It was a routine diagnostic lab. So everything was done with these ABI strips and looking for fermentation and color changes. And color changes I understood, and then I put my little code in, and then it says, ooh, it's E. coli. And so I liked this a lot. And the attending was very, very nice and said, well, if you're good at this and you have fun with this, why don't you stay in the lab and skip the sampling? And so I ended up in the lab. And I did this for 18 months, I think, at the time total. And at the end of the 18 months, I was involved in all the clinical. They have something called a ring test, basically a quantitation assurance system. And so I got all the unknown samples, and I could play with them and figure out what was in there. And so I fell in love with bacteria.
So you said
that's how it started.
You went to do a PhD in Berlin.
Yes. So I applied for medical school and applied for biochemistry grad program, and I couldn't decide which to take. And in the end, I took the biochem program and went to Berlin and started there. And then after a while, it was a little bit too narrow for me. So I like bigger pictures usually. And so I started to go to medical school in parallel. And
So you have an MD and a PhD?
Yeah, I actually have 2 PhDs and an MD, and the third PhD happened a little bit later when I started writing a book about filoviruses. And it started out as a review on filoviruses, it became bigger and bigger, and it became more interesting. And then finally it was a book, and one of my thesis advisor for my MD dissertation. So in Germany, you need to write a dissertation to graduate basically from doctor to MD. Said, well, why don't you pursue this further and actually make a thesis out of this in medical sciences, medical history? And I said, sure. Wow. If you support me, let's do that. And
3 PhDs.
So that's how I ended up with
Yeah, I noticed the 2 PhDs after your name, but
I thought it was a typo.
No, it's 2. It's 2 PhDs and 1 MD. That's enough. My mom says if I do one more, then she will slap me.
So it's Dr. Kuhn.
In Germany, theoretically, I guess, yeah, which of course is ridiculous.
If we were a professor, it would be even longer.
But yeah, Herr Doctor, Professor.
Wow.
Yes, it's a little bit bizarre how the Germans address each other. I have to admit this.
Did you do a postdoc at any point?
Yeah, I did a postdoc. So it's yes and no. So I did a postdoc / thesis at Harvard. So I had my MD already and then did my PhD. And so I was hired as a postdoc at the New England Primate Research Center under Mike Farson. And that's where I did all my work on, we tried to identify Ebola and Marburg receptor at the time and ended up identifying receptor binding domains and these kind of things.
Mm-hmm. And after that, you went to Frederick?
And then after that, I got a job offer and then I went to Frederick. And I was in Frederick before. So in Germany, when you go for a PhD, you have to have a master's. Mm-hmm. And so I did my master's thesis actually at USAMRIID, the United States Army Medical Research Institute of Infectious Diseases, which is right next door of our institute here at Fort Detrick. Mm-hmm. And that was in 1998. So I worked there for a year. And so now I'm pretty much on familiar ground.
Great. Well, we'd like to talk to you about Ebola classification, the current outbreak, and what you do. But we have a few things before that. We have some follow-up from last time. So, Kathy, we had a little email conversation, and you had mentioned that there was actually another paper on Relenza. Last time we talked about the Tamiflu Cochrane report. There's one on Relenza.
And during the show, because one of the summaries that I had read was something that talked a lot about the Relenza results. So it was a little confusing. And when I went to try and find the diagram that you and Alan referred to, that was the who knew what when the show notes came out on the TWiV webpage, I kind of followed my nose around from that. And then I found that indeed there was another full-length paper in the same issue about Relenza. And the results, in a lot of ways, are pretty similar. It was the same group of authors, but in a different order. They had looked at 150,000 pages of randomized controlled trials and regulatory comments and so forth. And what they found for Relenza was that it reduces the time to symptomatic improvement in adults, but not children. But the effect is attenuated by symptom relief medication. They found fewer harmful effects, evidently, mostly bronchospasm. Relenza doesn't reduce the risk of reported or confirmed pneumonia, and there wasn't any accessible information about hospitalization.
So did they have to wrestle the data from the... That's a different company now, right? It's not Roche.
Believe it might have been a different company, yes. And so I didn't follow up on how hard they had to work to get the data.
Yeah, it is... I'm looking it up right.
It's Glaxo.
Glaxo?
Glaxo, yeah.
It would be really weird if they had to wrestle it from both of them, and it's already weird that... So is it... You said it reduces the time to symptoms, so the Tamiflu was like hardly any time at all, less than a day, if I remember.
Right. This was mean difference in time to first alleviation of symptoms in days was 0.6.
Yeah.
So it's basically the same story. They're not terribly effective.
Right.
Yeah.
Okay. Wow. Well, we got actually an email from Stephen who sent a letter. This sounds like it's right up the TWiV comment alley. Stars include the Cochrane collaboration and Ben Goldacre, and it is an article about this story that we talked about in Night Science Journalism. I guess it's called Tracker by Night Science Journalism at MIT. And the title is "The Guardian Roche broke no law by withholding info showing its Tamiflu doesn't work." So it's basically saying, it's reviewing what we talked about last time, but basically by withholding the data, they didn't break any laws.
Right.
Which I had asked last time, could someone sue them? But apparently not. And this could happen in the future.
And it could be ongoing.
Yeah.
And I think that's the point that Goldacre was making in his article, which is that the data for most of the approved drugs, you've got lots and lots of data that have never seen the light of day that are being held by the companies. And of course, they're going to put their best data forward to the regulatory agencies. So how do we know that the drugs work that governments and insurance companies are spending billions and billions of dollars on?
We need to have a little reform here, right?
That's certainly what Goldacre would argue, and I would agree with that part.
So the world has spent $9 billion stockpiling Tamiflu.
Right.
How many R01s is that?
Yeah.
Wow. Anyway, thanks for that, Stephen. Another Stephen, maybe the same, or writes, "Dear TWiVsters, I want to give my reactions to the last TWiV in the discussion of reforming science. First, as someone who spent time in the humanities, we had similar problems accepting too many graduate students, though we were more concerned to make sure everyone could be paid throughout their studies. But in other ways, we were luckier than the sciences. I almost feel privileged now that my department got free cleaning, free electricity, even free office space, and salaries came from the university. Okay, maybe job prospects were far worse, but that comparison just makes much of what grants are required to pay sound even more absurd. Second, as someone with a number of friends who are young scientists, it seems like the role of staff scientists in science reform deserve more coverage. It's easy to see this as a less wanted career path, especially when, as you discussed, many established PIs are afraid of their domains shrinking. But I know some young scientists, and I doubt they're outliers, who would probably happily avoid the traditional PI career path in favor of staff scientist-type positions where they can spend more time at the bench and less time worried about grants, avoiding the bait and switch of science education. From what I hear, These positions are rarely advertised. They're more likely to be offered to postdocs who have been around a while rather than to people specifically motivated to have that kind of position. It's warming up here after a cool and overcast week, should reach 24 degrees tomorrow. More importantly, wind is abating. It was regularly gusting over 30 miles per hour earlier this week. Well, I don't know where here is. I have no clue. Could be anywhere in the world. Hey, that's a good point. The staff scientist, if we get to switching to that trend, I think it needs to be advertised. Obviously, you can't just give it to a postdoc. Yeah.
Well, a lot of job listings that you see in the back of Science and Nature are actually that, and you can sometimes identify them if they're hyper specific about the skills.
Yeah.
Because universities in many cases are required to advertise the job, even if they've already picked somebody who's going to get it. So what they'll do is they'll come up with an ad that describes that person, and put it out there and say, "Oh, well, this was the most qualified candidate."
Yes.
Yes, those jobs are sometimes advertised, but they're not really available.
Yeah, and then you apply for them and you never hear.
Right.
But they might be available, but people just don't know about them at all. So the problem, and that's one of the things that we see here with the different contracting companies. If there are positions open, then these positions are posted on the company website. So they're not necessarily going to the science or microbe classifieds.
Yeah.
And they're certainly not going to be posted on any blackboards at the university. So many people don't see this at all. If you don't know that the big players in contracting world are SAIC or Lockheed Martin or Battelle and so on, and you don't go on these sites, then you don't see what's there.
Yep. Last follow-up is from Ben, probably not Ben Goldacre. Hi, TWiV. Many thanks again for your amazing podcast. I've been with you since the beginning. Definitely not Ben Goldacre. And have written in a few times. I'm looking forward to more urban agriculture episodes as well. The first one was amazing. First, it sounds like several of you may be infected with the love of flying, which can be quite expensive. When I was in college, I learned to enjoy cigarettes. Then I developed a taste for cigars beyond my budget, thus losing my appreciation for cigarettes as well as my ability to finance my tobacco habit. I thus lost my tobacco habit. As the years have gone by, I've come to be grateful for that. No, I now believe I'm fortunate to have a variant of the nicotine receptor that renders me less susceptible to nicotine addiction than many, so your mileage may vary. Well, I fell in love with helicopters. I find those subtle birds far more fascinating than fixed-wing aircraft. They are also much more expensive and in many contexts less practical. I have to date indulged in only one lesson. I think they may play a role for me analogous to cigars. So anyhow, if any of you find your addiction to flight getting the better of you, then I suggest you give helicopters a spin. That's great. Very nice. I was just listening to some back issues of the Journal of Immunology's podcast when I came to realize that PTX3, secreted by murine and human immune and respiratory epithelial cells, is a competitive inhibitor of influenza hemagglutinin. This made me realize that any time we consider a drug target, we should first ask if the idea has perhaps already occurred to evolution. Our medicine should, I imagine, aim to complement those tricks which evolution has already considered. If we instead do something redundant, I imagine the in vitro results might look good, yet the in vivo results may disappoint, even with good PK pharmacokinetics, simply because the typical untreated virulence is already being moderated by the strategy that our treatment is pursuing. Supposing the PK of Tamiflu is good, and that seems like such a basic issue that I'd certainly hope it was confirmed early in development, perhaps the reason Tamiflu doesn't work is due to redundancy with some not yet discovered trick of our innate immune system. Are there any general lab or bioinformatics strategies for detecting such accidental redundancy short of large-scale clinical trials powered to confirm efficacy? I bet this is a common problem. In general, when it comes to any medical intervention, I figure we should modestly start with the question of why we think we know better than evolution. I think sometimes we really do, but very often I think we don't. This is a philosophy, however, that I don't often hear. Thoughts? I think we try to learn from systems, but I don't know if it's, you often say we're going to study innate immunity to identify drug targets, but I don't know that anyone really does that. I think they just run drug screens.
Yeah, or they come up with some molecule that's likely to be a good target and then try and design something in the case of hemagglutinin. you look at the structure, you say, what could we get that would bind to that and stop it from working, and then go from there without paying a whole lot of attention to how the cells are handling it.
Yeah, I mean, you mean neuraminidase and sialic acid?
Neuraminidase, sorry.
Yeah, I mean, there's certainly, this is a good point. There are likely many things that bind to our targets, but I do not think that drives discovery. What do you think, Kathy?
Well, I was thinking off on a different tangent about his question of why we think we know better than evolution. Mm-hmm. And I was thinking about how in my class, I told them about the pox virus story in Australia and to try and control the rabbits. And did we learn a lesson from this? Evidently not, because they're going in with another virus to try and take out the rabbits.
Yes.
So we still think we know better than evolution, and at least some of us do.
No, I think that this was very clear during the H5N1 debacle that everyone's saying, oh, we're going to make deadly viruses, but I think nature makes the most deadly viruses. They're pretty good at it.
Yeah.
And then some arise that aren't so good. As Ian Lipkin said, MERS coronavirus is pretty wimpy. It's not really going anywhere.
Oh, but increasing cases lately.
Yeah, I don't know why. Maybe it's camel season. I don't know why that would be,
they should take up cigars to quit the camels.
It could be.
Maybe it's more people reporting sick. Maybe it's better surveillance.
Yeah.
Maybe it's
lots of things. But it's still confined to the camel raising regions. All right, let's talk with you a bit, Jen. Hey, Jens, can you give us the an overall history of Ebola virus? We haven't actually talked about it very much here, have we, guys? Right? We haven't done an Ebola episode. In fact, Jens, I've been corresponding with you for over a year, and you said at one point, I could come on and talk about Ebola. So here you are.
All right. So let's expand it to filoviruses, if that's okay.
Sure, that's fine. And then then you have to explain the taxonomy, obviously.
That's right. We're going to go there. Yeah. So, the very brief history is that in 1967, an outbreak of something unknown happened in Marburg in Germany. Hence the name Marburg virus that was later discovered. And that happened among workers in a vaccine factory that received monkeys from Uganda. And at that time, there was not much personal protective equipment in the lab, so people didn't wear gloves, and accidents happened with scalpels and so on. And so a whole bunch of people became very sick, and many of them started dying. And of course, were admitted to the local hospitals, and the public health authorities called alarm. And it turned out that there was a virus in these imported monkeys, and it looked like no virus ever before because it was a filamentous virus. And until that time, and even today, filamentous viruses are usually, you find them in the plant, in plants, and you find them among bacteria, but not really among mammals. I mean, influenza virus is kind of a filamentous virus, and there are a few things that, depending on the nucleocapsid, you can kind of count filamentous, but nothing is there in the typical long spaghetti shape of Marburg virus and all the other filoviruses. So it was pretty clear pretty quickly that this was something new. It received the name Marburg virus. And then it disappeared again. So, I mean, there were a couple of single cases that showed up here and there in Africa later, but pretty much this was an exotic thing. And after the initial excitement about the virus, it disappeared. And then in 1976, there were two outbreaks in Zaire, which is today the Democratic Republic of the Congo, and in Sudan, which is today South Sudan, among several hundred people, where people get pretty much the same symptoms, first flu-like symptoms, and then more and more severe diarrhea and vomiting and eventual hemorrhagic symptoms, and with a very high case fatality rate. And people were very quick in assuming that this could be Marburg virus because of those symptoms, and also because they found these spaghetti shapes. And further studies then revealed that these viruses were not Marburg, they were something related. So they called these viruses Ebola virus. And it continued for a few more years, and a few labs in the world worked on this, mainly CDC, Porton Down in England, and USAMRIID, of course, here next door, started working on these viruses, characterizing them a little bit antigenically. And so it turned out pretty quickly that the virus that was causing the outbreak in Zaire is a different virus than the virus that caused the outbreak in South Sudan. So now we know these two viruses as separate viruses, as Ebola virus, that's the one from Zaire, and as Sudan virus, that's the one from Sudan. And then one after another, more of these family members popped up. So there was Reston virus in 1989, showed up in Reston in the US after monkeys were imported from the Philippines. Then TWiV virus, which was discovered in Cote d'Ivoire in 1994. And then Bundibugyo virus, which was discovered 2007, I want to say, in Uganda. So now we have 5 different viruses: Ebola virus, Sudan, Reston, TWiV, and what am I missing? Bundibugyo. And we have Marburg virus and a closely related virus that was discovered in 1987 in Kitum Cave, which is now called Raven virus. And so if you do whole genome analysis and the usual alignments and everything else, you see that these viruses cluster into groups. These 5 Ebola-like viruses we now call Ebola viruses. And then the 2 Marburg-like viruses, Marburg and Raven, we call Marburg viruses. And both these groups together form the Filoviridae family. And the name Filoviridae comes from the filamentous appearance of virions and when you do EM staining.
So the family is Filoviridae.
Correct.
And then there's
a genus. There's a genus called Ebola virus,
which is one word capitalized, right?
That's right, which is one word capitalized. And so here it gets a little bit complicated for reasons that are complicated. But so we have an Ebola virus, which is one of 5 Ebola viruses. And so this is complicated when you speak to differentiate. I mean, I could point out that there's a differentiation in pronunciation if you want to be really nitpicky and geeky. So the actual virus from Zaire, the pronunciation should be closer to Ebola virus, because it's a French word and has an accent aigu on the E. So if you listen to talks about this virus by French, Germans, or Russian scientists, they pronounce it usually correctly, and Americans and British usually make an Ebola out of it and put the stress on the wrong place. So we tried to come up with the genus name in a way that you can at least differentiate this. So I should say Ebola virus is one of the Ebola viruses. And I'll try my very best to keep this up during the conversation today. So then there's a second genus, Marburg virus, and here we have the same problem. So the original Marburg virus is one of 2 Marburg viruses, the other being Raven. And then there's a third genus that was just accepted last month called TWiV virus, and this is for a Ebola-like agent that was discovered in among bats that were sick in a cave in Spain, and that virus is called Yovio virus.
Okay, so the way the press does it, they call it Ebola, capital E, separate from virus. That's correct now, right?
That's correct for the one that, for example, is in Guinea right now, yes.
But it's not correct if you use it to refer to all of the Ebola viruses, right?
Right.
All right, and that's
it's a little bit like in all the other viruses. I mean, you don't, like you have the arenaviruses, and within the arenaviruses you have Lassa and Majupo. So you don't say that's arenavirus Lassa subtype. You say it's Lassa virus. The only unfortunate thing is that we have the same word stem both in the virus as well as in the genus. And so I am in the ICTV Filoviridae study group, and we are trying to find a solution to this, but it's not simple to do because we are trying to establish taxonomies, first of all, prospectively, to deal with new viruses that are going to come sooner or later through all the deep sequencing efforts, in a way that is both database compliant and ICTV compliant.
So the other viruses are called Reston virus, Sudan virus,
Bundibugyo virus, TWiV virus, Raven virus.
All right. So you don't. So the press would probably put Ebola in there somewhere, right?
Right, because it just sounds more exciting.
Yeah. Okay. In terms of outbreaks, how many cases have there been of Marburg and Ebola viruses using the genera?
Well, since 1967, there have been, excluding the current outbreak in Guinea, because those case numbers are not firm yet, but since 1967, there have been about 2,800 infections, human infections, and about 1,940 deaths. So 2,800 cases.
1,940, okay. Okay.
And that's infections counted as cases.
Yes. So there are some papers out there that suggest that there might be subclinical infections, but I don't think it's generally accepted.
Really?
And there's very little evidence. So if you get infected with one of those viruses, I would say you will get sick. So the cases that we know about are people that were sick.
So there's a study I looked at not too long ago from 2010, 4,300 people in Gabon.
Mm-hmm.
And they found antibodies against Zaire virus in 15% of them. So you don't buy that, you think? You think the serology is off?
Well, it's very complicated. It is. Serological studies have been done, of course, from the very beginning since these viruses were discovered.
Yeah.
And the puzzling thing was that pretty much anywhere where people looked, and in the beginning these were all IFA studies, they found antibodies against Ebola viruses,
Mm-hmm.
not against Marburg viruses. Usually Marburg viruses was in the lower 1% range in their sampling. But for Ebola viruses, they could find 10, 12% seropositivity in a lot of different countries, and not only in Africa, but also in European countries. Even in Alaska, they did a study and found 2% or something.
Yeah.
So that told people pretty quickly that whatever antibodies they had against Ebola virus were probably cross-reactive against something else. So
I have a question before you go on. So I wasn't sure which Ebola virus we were talking about here, whether it was the genus or not. So the
in the beginning, everything in the beginning was done basically with the antigens of those two viruses that were discovered in Zaire and in Sudan.
Okay.
So most of the serological, because the later viruses were only in 89 and 94, and by that time a lot of the serological studies have already been done. So they were done with those two antigens.
Okay.
So pretty much until until the discovery of yo-yo virus, most of the people in the field discounted all of these IFA studies as complete bogus, except of course the people who did those studies. And then it switched a little bit because people developed a little bit more stringent criteria on what a serological study should do. So they switched away from IFA towards ELISA. They were looking for confirmatory Reston blots. And then, of course, slowly with time, confirmatory PCR, trying to find fragments and so on. So then it got a little bit more interesting because studies were repeated. And so the study that you looked at probably was one of those ELISA Reston blot studies. And probably this can be taken more seriously in the terms of, I would buy that the people who have positive responses probably have been in touch with some kind of a filovirus.
Mm-hmm.
I'm not really sure though whether it's one of those really evil ones.
Yeah.
And so that's why I brought up Yovio virus, because that's the virus from Spain. There were of course no human infections or cases or anything in Spain. This was a random finding. Looking at bat die-offs in a cave with deep sequencing, and this genome popped up, which is clearly a filovirus. But it was puzzling because the caves over there, they're very popular tourist goals.
Mm-hmm.
And so there were a lot of people in these caves, and it was a little bit curious that nobody got infected. And Reston, of course, is a virus that is kind of considered non-pathogenic by most of the people. And we don't know of any human infections of this virus either. So now you have two viruses in this family that could be around and you could be exposed to it, but they don't make you sick. And so if that is the case, then I wouldn't be surprised if there are many more filoviruses out there that we haven't seen yet, and in some funky reservoir somewhere. And maybe you're exposed to that, and then you have cross-reactive.
All right, but you think that if you get infected with Zaire or any of these Sudan, if you're going to get sick, you
Yes.
get you have a high likelihood of dying, right?
Yes.
Okay.
So I mean, the case fatalities are not quite as high as the press likes to make them, and also the scientists like to make them in their first intro paragraphs and papers. They usually choose the most horrendous outbreak, and so in terms of the virus from Zaire, Ebola virus, they usually write case fatalities up to 90%.
Yep.
Which of course is not a wrong sentence, but it's also not a right sentence. So I mean, there have been outbreaks with small case numbers that have been 100%. Like 3 cases, 3 people die, and there have been outbreaks with 1 case that survived. So you have the entire spectrum from 0 to 100%.
I mean, it would be interesting in this current Gabon outbreak whether if they did some serology on the surrounding population to get a sense of whether there was more, because they postulate that the virus has been around since December of last year. So you
mean Guinea, right?
Sorry, Guinea. Yeah.
Yes. Mm-hmm.
So you would think that it has infected other people, right? Without
most likely yes. So I think with many of these countries, you have a lot of diseases and a lot of agents endemic that cause death. And if you look at Guinea, for example, malaria is very prevalent and causes a lot of deaths. So in the beginning of the disease, it's not easy to differentiate just by clinical presentation a filovirus infection from malaria or a whole bunch of other diseases.
Yeah.
So if the healthcare system is not completely vigilant, people will be put in a hospital bed and they might die of something that you don't know they have. And because filoviruses are not very contagious, contrary to the Richard Preston kind of scenario, it doesn't necessarily mean that one person who lays there needs to infect many other people.
Yeah.
It depends on how he's being treated and which kind of PPE is in place. And if you look at the Marburg virus infections, those single ones, people went on a hunting trip or on a hiking trip, and they came back with that disease. And because they didn't encounter anybody or they didn't have direct physical contact with people, other people did not get infected, and you had a single case. So it's very possible that this virus, the study that was published by Günther, showing that this virus in Guinea is not a virus that just wandered from Democratic Republic of Congo or Gabon into Guinea, but actually has been there for quite some while. That seems very plausible. There were studies done by Townsend Peterson in probably beginning of 2000 or so. They did ecological niche modeling. And they predicted that Ebola viruses are present in West Africa. They predicted that Marburg viruses are present in a whole bunch of countries where we haven't seen them yet, just by vegetation and looking which kind of territory there is.
In any of the outbreaks, including this current one in Guinea, do we know the source of infection?
It's complicated again. For the Ebola viruses, we really don't know. So if you want, you can speculate and you can make some good guesses, and then of course there's data. So let's start with data first. So for Sudan, for Bundi-Butchu, we have absolutely no clue where they're coming from. You can only do the typical histories of patients where you say, well, what did you do? And then they will tell you, well, I was hunting and I found the following dead animal in the woods and I hunted the following monkey and we ate that monkey. And because there's a certain pattern with Marburg virus being introduced into Germany through non-human primates that were imported, people jump to conclusions very quickly. It must be a non-human primate. For TWiV forest virus, which caused one human infection thus far in Côte d'Ivoire, we know where it's coming from because there was an episodic among chimpanzees that was studied by zoologists, and the person who performed the necropsy on one of those chimpanzees got infected. So chimpanzees somehow, at least once, were in touch with that particular virus and got it from somewhere. But because those animals were also so sick and they all died, they were certainly not the host. For Ebola virus and for Reston virus, Reston virus is a very complicated story because it's the only virus that thus far has not been seen in Africa, but seems to somehow come from the Philippines. And it was imported by primates, but I don't think that anybody thinks that these primates were necessarily the natural reservoir or in touch actually with the natural reservoir. It looks more like they were in touch with something during captivity. So that leaves Ebola. And for Ebola, there are some hints, mainly in terms of bats. So there were a few studies done where antibodies against the virus were detected in bats and in the same bat population, and genomic fragments were detected by PCR, but never antibodies together with genomic fragments in the same bat. And so the common idea there was that bats somehow get infected, they might clear the infection after a certain time, and so depending on when you sample, you find still the genome or you find the immune response and find the antibodies. The story is different from Marburg and Ravin. Those two have actually been isolated from bats, from Egyptian rosats. Those are fruit bats. And so with this, I would say, okay, we definitely know that these bats get infected naturally in some kind of a way. And these bats live in caves. And these caves have been frequented by people who got infected. And so there is a logical chain of transmission that is not proven, but at least logical. People went in there, cut themselves, got exposed to guano, or maybe it's little particles in the air or something that they inhaled or something like this. And they found viruses in those caves where cases were recorded. So it seems logical. I'm still a little bit skeptical because the overall infection rate in these bat populations is relatively low. And so I don't know exactly what that means. So if you compare this with hantaviruses or arenaviruses, if you sample rats in Nigeria and you look for arenaviruses, you will find them pretty much everywhere. That's not really the case with Marburg and Raven in bat populations. And also these bats of this particular species, so that's Rosetus aegyptiacus, are distributed over a whole much bigger area than we know that there are cases. So there's still a riddle about, could it be something else that introduces the virus into the bats and then you get it from there? And the other question, of course, if non-human primates, hunting of non-human primates or finding dead non-human primates in forests are the main reason why people get infected, then how does the virus travel from the bats to the non-human primates? And so there's a whole bunch of open questions, I think.
Maybe it's the camels.
Yes, so far, no, I haven't seen anything in relation to camels with filoviruses, except, of course, that that people use their antibodies now for diagnostic acid.
Right.
Is it correct that some of these viruses are in gorillas?
Oh, that's a loaded question. So my friend Peter Walsh and I would start fighting immediately. So Peter and of course a whole bunch of other people have done very interesting studies that statistically show that gorillas, especially gorilla populations, decline at the same time when there is an outbreak of Ebola virus among humans in the same area. And so that statistical correlation is very, very strong.
Is that because they stopped hunting chimpanzees and started hunting gorillas?
That's an interesting question. I don't think they hunt gorillas, so first of all, they're different animals that are being mentioned with these with this kind of eradication by Ebola. And so if you look into the press, you see duikers and chimpanzees. The duikers are small forest antelopes, duikers and chimpanzees and gorillas. And I would say the data set is the weakest for the duikers because, as far as I remember, only antibodies have been found in duikers. And as I mentioned before, antibody finding is always complicated. Antibodies have also been found in dogs and in rats and, all over the place. Then there are chimpanzees and gorillas. And the chimpanzee populations, as far as I have seen in papers, the statistical significance in terms of die-offs is not nearly as strong as with gorillas. So that leaves the gorillas. And in the gorillas, what they found were in a handful of animals, 5 or 7 or something like this. They found fragments of the GP gene of Ebola virus.
But a handful of gorillas is a lot.
Well, yes, because of course it's very difficult to sample them.
Yeah.
So this is a big problem. Of course, and that's also one of the main arguments. I'm kind of one of these people who say okay, filoviruses grow very readily and happily in simple viral cells. So I would like to see an isolate from a gorilla to buy this.
Right.
And Peter would immediately say, you don't need an isolate because, first of all, the statistics are very good. And second of all, we will never get this because you will not find a dead gorilla in the forest because they decompose so quickly or get eaten by other things that the likelihood that you stumble through the forest and you find some animal that just died 24 hours ago and still has tissue that you can use for isolation or full genome analysis is basically zero. I would say, I give him that, but I still would like to see that isolate.
I think that's a good standard to be held to. You don't want to change that's what we do in virology for years, right? You want to get viruses. That's what we gave Ian Lipkin a hard time for about the camel coronavirus story.
Right.
Now, they had 190 bases initially and then some antibodies, but they eventually got the virus out.
Yeah, I think, I see from a taxonomy point of view, I see both sides. Yeah. So I think, we know things about biology of certain viruses. And as I said, filoviruses are relatively easy to grow. All of them that we know are relatively easy to grow. So I don't see why it should be hard to find these viruses.
Yeah.
It's a different story when you think about hepatitis C or these kind of things.
Sure.
Where I wouldn't discount the existence of a virus just because there is no isolate. And so this is another fight that is happening among people who do taxonomy. I think if the coverage is good, if you use high-quality deep sequencing and you find a full genome, I would believe you that that virus has been there. And I might actually believe this for the filoviruses too. If you present me with a full genome with good coverage in a gorilla, I would buy that too. But a simple PCR where you can find a consensus sequence and you don't even have an entire gene, but only pieces of it, I'm getting suspicious. It could be anything. First of all, they could simply be exposed in some kind of a way and has no effect on them, and you pick the right time. But then also, cross-contamination with your positive control, all of these possibility. So I would like to see some variation. The sequences need to somehow be convincing that this is really that way.
So I announced on Twitter today that you were going to be here, and some people sent some questions. One of them is from Amboceptor. It says, is Ebola virus inherently rural because of proximity to animal reservoirs? If not, what cities are most at risk?
Oh, it's a good question. So traditionally, I would say that if you look back at most of the outbreaks were in fact rural for most of the filoviruses, but there have been important exceptions. So I, again, extrapolate to filoviruses, not only Ebola, but Marburg, of course, was in Angola in an urban area. Ebola right now in Conakry and is also in an urban area. So I think, yes, of course, it has something to do with the host. And it has something to do with the overall status of the country in terms of wealth and public health. So if you have a city that basically has no public health infrastructure and no hospitals and no doctors available and no PPE, then of course you will have an explosion the moment the virus gets introduced. But it raises a different interesting question. So, I mean, I told you there were 2,800 cases total since 1967. That's a very small number in the first place compared to other diseases. But I think what people don't really appreciate is that most of these outbreaks could be traced back to a single index case. And so this is the same thing right now with the outbreak in Guinea. That paper that was published by Stefan Günther shows very clearly that at least the entire first transmission chain is going back to, I think it was a little girl that got infected and died.
Yep.
And so that's the case for pretty much every outbreak, with the exception of one Marburg outbreak in Democratic Republic of Congo, where there were several introductions. And that, if you count then these outbreaks, you realize that there have only been about 50 introductions of filoviruses into the human community since 1967. That's an incredibly small number. And to me, that means that, something extraordinary has to happen that you get these viruses. So either the host never sees humans, but then because we encroach it in some kind of a way, there's some kind of a reaction where it bites us, although it usually wouldn't bite us. Or there's some kind of a ritual that only certain people do with certain animals. Or it has to be something a little bit more complicated than I was exposed to bats.
So those are 50 introductions that we know of, but there could have been more, right?
Correct. So based on the fact that we know of, there's no real definite proof of subclinical infections, I would say that is true. It's of course possible that there are subclinical infections. We have not detected them. And then that entire story might change.
Well, for HIV, there were at least for HIV-1, there were probably 4 introductions, right? That led to the 4 groups, MNOP. But there may have been many more that never went anywhere.
Correct.
Because you do get apparent infections.
Yes, of course. And as I said before, if a single person gets sick somewhere on a trip in the forest and then dies somewhere, we will never know about it, and it could have happened many times. Yeah,
this paper, it's in New England Journal that you mentioned by Günther. They have this wonderful figure where they show the chart. They did wonderful epidemiology of all the cases, and you're right, it traced back to a 2-year-old child who died in December of 2013.
Yeah, and if you go back to the old outbreaks, first in 1976, and also the Sudan outbreak, there are beautiful personal accounts of people who were involved with this, where you can see this over 318 patients, how everybody was connected with everybody.
It's incredible. It's like her family got sick, and then there was a midwife who got it and spread it to others. Oh man, it's just
and it's a very typical story for these viruses, because the first thing that happens when somebody gets sick is, of course, that the family tends to them. And they have a fever, so you start with cold water and cloth, and so you touch each other and you caress each other, and unfortunately this virus, that's the way it spreads.
Is this outbreak ongoing still?
It's a good question. So the last reports that I saw was that there were no new cases, I think, for like 3 or 4 days or so. But there's still some people in treatment, at least in Guinea. And of course, 2 incubation periods have to pass before somebody can declare it over. So I wouldn't say it's over yet.
Right, and the incubation period can be a few weeks for this, right?
The standard rule, I think, that WHO issued is 21 days.
Okay.
What kind of lab tests and facilities do you need to have to confirm a diagnosis?
Well, the standard diagnosis right now is a combination of, first of all, clinical presentation and epidemiology combined with usually antigen capture ELISA and PCR. So the good news about that is that you can do this as long as you have the necessary technology at hand and electricity and so on. You can do this pretty much anywhere. There are ways to inactivate samples from patients, many different ways, that inactivate these viruses very rapidly. I mean, these are negative strand RNA viruses that are enveloped. So this is also something that people need to remember. Disinfection is very easy with soap, destroys the envelope and the virus is dead. Heat destroys it. Sunlight destroys these viruses. So that's it's easy to relatively easy once the education comes through to quarantine patients and quarantine the outbreak. And for the same reason, you can treat samples to do diagnostic tests. But eventually, of course, these samples are being sent to a high security lab somewhere, a BSL-4 lab, either in the region, so in Africa, that would be in Gabon, which has a BSL-4 lab in Franceville, or to Johannesburg in South Africa. Or, of course, in this case, in the Guinea outbreak, they were sent, I think, to Hamburg and to Lyon, so Hamburg in Germany, Lyon in France. And if the outbreak is in Uganda or other countries that have close ties with the US, they would come to CDC or USAMRIID. And then these labs would, first of all, confirm the diagnosis with their own tests, and then, of course, attempt virus isolation and sequencing.
So I misspoke earlier. I had suggested that this virus had been circulating unknown in December, but in fact, that's when the first case was in December, yes. The little girl. So it has been around since then, and she must have. She obviously acquired it from some source, and we probably can't tell because she's only 2 years old and couldn't do any history before she died. It's also not easy to do these interviews. So it depends on which country you're in. I mean, this is many people just subsumize Africa a little bit too easily, and of course there are vast differences in culture and belief between different countries. And so performing an interview is not necessarily easy depending on the language that people speak. This is actually something I'm working on. I have a linguist on staff, and we're looking a little bit into this. Most of the countries, of course, have official languages. So if you go into the Democratic Republic of Congo, French is the official language. But of course, that doesn't mean that all the people in the country speak French or speak French to a level that allows them to talk about medical circumstances.
Or would be willing to talk to somebody who seems like they might have come from the government.
Correct. For the government, or come in suits that are threatening to them, or simply, like there are still colonial fears in many of these countries. So simply being white can be a problem as well. People are afraid, and if you look at the history, justifiably so.
Yeah.
So they would not necessarily divulge all the information, and there is, of course, a tendency of people from the West to look at rituals and religions that they don't usually have or are not exposed to laugh about them, which is a big problem because, of course, then nobody will tell you about their beliefs and why they did certain rituals and whatnot. They will simply keep it to themselves, and that's one of the reasons why CDC and WHO started about, I think, 10 years ago to involve medical anthropologists in outbreak control to raise the sensibilities among outbreak personnel, control personnel, on how to talk with each other and how to educate each other. And also, of course, not to come across as something imperial or better.
Right.
So I have a question because I haven't quite figured out how your work fits into this. And I'd also like to hear about the relationship or jurisdiction of other things like CDC, NIH, and so forth in this kind of outbreak, in addition to the international other countries that you mentioned.
Okay, so my work, let's start with my interest. So I got into all of this because I was interested in BSL-3 and BSL-4 viruses relatively quickly after I left the bacteria behind. And so I started working at Harvard in a BSL-3 lab on HIV in Joe Sadrowski's lab, because at that time HIV was mainly classified as a S2 plus or something agent, so people put them in BSL-3 labs. And then I started wandering around to BSL-3 labs and BSL-4 labs. And while I did my master's thesis, I got exposed to Ebola for the first time. That was at USAMRIID next door.
So to speak.
Yeah, not literally.
That's right, not so to speak. That's right, so to speak. To the topic of Ebola virus. So I worked on development of vaccine candidates at USAMRIID for a year as my master's thesis. And, you know how this goes when you have students who are writing master's thesis. Eventually, they need to start writing something and need to come up with an introduction. And so I started looking at papers out there and started to summarize these things. And I quickly realized that the summaries of the filovirus field in the publications that you find on, well, at that time I didn't even know what PubMed was, I think, but on general papers that were around, I found them incredibly biased. And I guess that was my German heritage, because I told you that Marburg virus was discovered in Marburg in Germany. And so, of course, the first 20 or 30 important papers on Marburg virus were published in German journals and in German, usually in the Deutsche Medizinische Wochenschrift, which is German Medical Monthly. But nobody cited those. So everybody cited the science paper from Kissling et al. From Porton Down, which was published a couple of years later, because of course we like science, sounds good. And so I found this kind of wrong. And I do think that everybody, no matter where you work and in which conditions, but if you discovered something, you should get the credit. And I always correct this in peer review when somebody says this virus was discovered in 1920, and then I see a 2002 reference that bothers me. So I thought, well, okay, for my master's thesis, I'm going to write a little bit of a better review. And I started to dig around and try to find the right journals. And so I found the German articles, and then I quickly stumbled upon the wealth of Russian publications that nobody ever cited or summarized, mainly because, of course, the people either didn't know they exist or because they didn't speak any Russian. I don't either, but I found people who helped me then at the time. And so that's what I mentioned before. This review got a little bit bigger, and soon it was not an introduction for the master's thesis anymore. So it was kind of a review that I could publish, and I continued digging more, and suddenly it was too big for a review, and it turned into that thesis. And because it was a thesis, and in the end it became a book that was published in 2008 that summarizes pretty much everything that was known about filoviruses until that date, including government reports and abstracts and theses and so on. Ebola and Marburg became somehow my baby. And so that's in the end why I continue working on it. I'm fascinated with the viruses. I'm fascinated with everything that's associated with it, the cultural impacts, and also the fascination of other people with the virus. I find quite interesting the sociological studies that are associated with it. And so from then on, my career was targeted towards places where I can work on these viruses in any kind of capacity. And so that's how I overall fit into the field, I guess. I'm not a field worker. I'm not a clinician who goes to Guinea and treats patients. That's what other people are doing. But our facility, the integrated research facility at TWiV where I work, is an NIH facility that will facilitate the development of medical countermeasures against high consequence pathogens, such as Ebola, Marburg, Lassa, Machupo, Nipah, all of these BSL-4 agents. So this is a very new facility. We have the state-of-the-art BSL-4 facility, of course. But we're also unique because we have medical imaging in the BSL-4. So that means we have SPECT and MRI and PET scans and so on in the BSL-4 suite. So that's unique worldwide. And the idea behind that is that we can not only learn new things about pathogenesis, of course, about these viruses, but we hopefully can evaluate the use or uselessness of medical countermeasures in real time without having to kill a lot of animals. So that's my other leg, so medical countermeasure development in that field that I'm interested in. And because there are only very few BSL-4 facilities in the world, there are probably about 12 or so that are of a reasonable size, and then a bunch of other small facilities that have a diagnostic capability. Whenever there's an outbreak, usually most of the facilities somehow get contacted and try to help mitigate the outbreak. And so in our case, our associate director, Lisa Hensley, she flew to Liberia, and she just came back actually, and assisted all the different organizations that were involved with containing the outbreak in Guinea. And so that's, of course, USAMRIID next door, then the CDC is involved, NAMRU, WHO, the different ministries of health of the different countries, and so on.
Do you think we need more BSL-4 facilities?
Oh, that's one of those questions that my boss is not supposed to hear, I guess. No, I actually don't think so. Maybe we need different ones. But I think, everything needs to be in relation to the actual threat. And of course, in the BSL-4 field, there's the one possibility, of course, that people get exposed to these viruses somewhere when they go somewhere, and we want, of course, to be able to treat them. and we want to help the countries that have the outbreaks. And so of course, if there's an outbreak in Guinea, ideally we would like to fly in and help everybody. But of course, the overall case numbers, as I said, especially for Ebola and Marburg, are relatively minuscule compared to, let's say, malaria or river blindness or other diseases that actually cause a lot of suffering in great numbers. At the same time, of course, there's the association with bioterrorism and biowarfare, and so the threat that somebody could use these viruses because they are so infectious and because they're so lethal for bioweapons development, and that these weapons could be deployed against anybody. And the question is, what do we do then? And so we need to be prepared. So there's a balance between these things, and I think we have reached that balance. We have, I think, enough BSL-4 facilities in the United States and also pretty much worldwide. It would probably be good to have a little bit more capacity in Africa, given that these outbreaks are very prevalent in Africa. But the collaboration seems to be going very well. Especially CDC has a lot of ties with all the individual local labs, and by now, as far as I can see from the outside, exchange of strains, exchange of clinical materials, and also exchange, of course, of countermeasures and treatments and diagnostic kits and so on is going pretty seamless and pretty fast.
So we don't need the needle in Boston?
Oh, that's a loaded question.
Yes, deliberately.
Yes, deliberately. Well, let's put it this way. The building is there. It is theoretically functional, and it would be a waste for science not to run it. I don't understand. I understand the general fears, of course, of the population that they don't want to have a facility in the middle of a city. This is because, of course, movies like Outbreak and Richard Preston's Hot Zone and all the other TV series that we see about these viruses somehow perpetuate that image that if there's a hole somewhere, Ebola will fly around and infect everybody, which, of course, is not true. So it's a mixture of, I think, misinformation in a way, and also bad handling. So I think both sides in this fight of whether this facility should open or not, especially in the beginning, are a little bit to blame of why we are in this situation right now where everything seems to be stuck. But from a scientific point of view, this is a fantastic state-of-the-art facility. It's very safe. The people who are working in there, I think you did a TWiV with Elke Mühlberger, I guess.
Mm-hmm. Yes.
Yeah, I mean, the people who are working there, especially Elke, they're very, very good. Have a lot of experience with these viruses, have, of course, a spotless track record in terms of accidents. There were never any. And to have this multi-million...
You mean at that facility?
Well, at that facility, but also like if you look at Ebola and Marburg virus laboratory accidents in general. The only ones that happened. There's
a little bit of complexity when you're talking about Boston University and bioaccidents.
Oh, yes. I'm talking about, whenever I talk about my BSL-4 virus.
Now you've zoomed out to BSL-4 in general, right?
That's right, yes. So, but in terms of BSL-4, the track record of BSL-4 research has been pretty much spectacular. If you discount the first, 10 years in the 60s or so when people didn't know what they were working with and how to work with it.
Well, but that wasn't really BSL-4, right?
Right. So they were like, high containment as whatever you could make as high containerish as possible at that time.
Right.
So there were different times. And so most of the laboratory accidents that happened with BSL-4 agents later happened in countries where the safety was not necessarily that good, or where people worked with quantities that were, outrageous. And so the example for that, of course, it was Soviet Union within the bioweapons program.
Yes, Sverdlovsk, right?
That's right.
So do you?
And you're referring to human BSL-4 facilities. There was a veterinary incident, but that's separate.
Yes, and of course, then you get into the what's the BSL-4 in Russia and what's not, and so on, and which agent is classified how, and it's all getting very messy then very quickly.
Do you work in the 4 over there, or are you?
No, we are actually only on the verge of being hot. So we have been competing with the needle.
Really?
So our facility is, we are finished, of course. So the entire building is built, and we are now in the BSL-4 labs doing research with non-select agents. Mm-hmm. But we just received our select agent clearance, as far as I heard from our leadership just recently, and we are now looking at how to obtain the agents and how to slowly start the program. But I have a lot of collaborations with Nextdoor, with USAMRIID. So when I'm a molecular biologist, so most of my things are in vitro surrogate systems. I work a lot with pseudotypes and VLPs and so on. And whenever we need confirmatory experiments with live viruses, then we usually collaborate right now with USAMRIID and Data.
So USAMRIID has a live BSL-4, obviously.
Yes.
Right? And yours is relatively new as well, right?
Oh, it's brand new. The building is, so when I started, I had my 6-year anniversary now at this institute, and when I started, this building didn't really exist. So I started in building 33 on the main campus at NIH until this building was built enough that I could pretend to have an office somewhere and move in. And in the beginning, everybody around me wore hard hats, and it has changed a lot. And now it's a completely finished, beautiful facility, actually.
Oh, so that means we could have visited it because it's not hot yet, right?
Yes, that's right.
I have a question from Welkin Johnson, who I guess you know. He says, hi to Jens. Jens, sorry. My questions are related to the prevailing view of Ebola virus outbreaks in people. The current model is spillover infections from a reservoir, followed by a rapid but self-limiting outbreak. How do we know there is not low-level persistence in a small fraction of people or asymptomatic carriers spreading virus subclinically? Maybe seemingly unrelated outbreaks are actually traced back to single spillover events. Are outbreaks immediate consequences of spillover, or could the spillover be temporally separated? Similarly, are there data on exposure versus outbreaks? Do most exposures result in outbreaks, or might most exposures be benign and go unnoticed or be misdiagnosed because they aren't severe?
Yeah, that's a typical Wilkin question. I worked with Wilkin, well, not with Wilkin, but next to Wilkin at the New England Primate Research Center when I did my postdoc over there. And so he was professor next door. So we had a lot of these discussions because he's a retrovirologist, and so he loves ecology, especially of endogenous retroviruses and all these restriction factors and so on. Well, the short answer is, well, we don't know. Because we don't have any data on subclinical infections other than 2-3 papers that looked at immune responses and so on. So I'm not aware of any studies that detected either like a virus, and we have virus isolation, or a full genome in a human that walked around happily. So I find it unlikely that the viruses that we know and that are classified are circulating around in a subclinical fashion. But as I said before with the Ovio and so on, I wouldn't be surprised if there are other filoviruses that don't cause anything and they're circulating, and maybe people do get infected with them and we simply don't know about it. Yeah. Now, in terms of the introduction, does every introduction lead to an epidemic? I would say no. I mean, first of all, if you look at the number of outbreaks and the case numbers of the individual outbreaks of all of these viruses, you find a lot of outbreaks that have very small case numbers. So just looking at Sudan virus, for example, so the first outbreak in 76 was a big outbreak with 284 cases. But the second one had 34 cases. The third one was very big with 425 cases. And then all the other outbreaks that came after had 17 cases, 1 case, 6 cases, 11 cases. And so this is true for the other viruses as well. So there's a big variability in terms of outbreak size. And this outbreak size is most likely determined by where the infection happens, how quick the patient develops clinical signs, and what he does after. So if you get it alone somewhere and you go home to your house wherever you live and you roll yourself up in bed and you die and you don't have a big family that is watching after you, then of course probably the outbreak is going to stay small. But if you don't know that you have something, but you are infectious and you go into a hospital where people take blood and reuse needles and go from one person to another, then of course you might have a big explosive outbreak. So I think it's very much dependent on country and within the country on where people live, whether it's a city or whether it's a village or whether it's a small little hut somewhere that's completely isolated.
And I assume there might also have been more of these smaller outbreaks further back in the past that just nobody noticed at all, right?
Yes, and I also think actually that this is probably true for today. So if you look at the Democratic Republic of Congo, it's a very big country. So with very varying infrastructure depending on the region that you go in, with ongoing military conflicts in different areas.
Right.
So if for most of these outbreaks, if you go into the past, they went unnoticed for a very long time before a local physician then finally called the government of that particular country, and then that government was willing to out loud say we have an Ebola virus outbreak, and then call help of WHO or somebody else. People don't want to announce these outbreaks. This is like similar if you go back to SARS and China and how long it took until somebody actually rang the alarm bells.
Well, in Saudi Arabia.
That's right. And so there's a question, of course, of how many small outbreaks simply go unnoticed, or actually are noticed by a local, but the local doesn't dare or think it's necessary to report it to somebody higher up.
I have a question as you might have guessed, Wilkins was not via Twitter. It was sent by email.
It's a lot of
Twitter.
tweets. Yeah.
One more from Somya Dip. He says, can Ebola be eradicated like polio? Well, we haven't eradicated
We don't know if
polio.
Ebola can. If polio
Yeah,
can be
I'm not
eradicated.
really sure whether polio will be eradicated, so we'll see about that. But it's a very hypothetical question, because as long as we don't know where these viruses hide,
Yeah.
it's possibly. If it's a very strange, it might eradicate itself. I mean, I told you that there were only 50 introductions in the human population. So maybe this, let's speculate out loud and be completely crazy. Maybe it's a mosquito that is endangered.
Mm-hmm.
And there's only a few mosquitoes left, and we encroach on the environment and destroy the habitat, and the mosquito disappears tomorrow. Then the virus would be gone. But on the other hand, the introductions into these bat populations shows that there seems to be some kind of ongoing contact, and if they can replicate in bats, then it's of course possibly that there's a species jump eventually manifests itself completely in bat populations. Then it's going to be a complete different game, because of course bats are inhabiting caves by the hundreds of thousands.
Well, why don't we speculate in the other direction, which is what the press likes to do? Is this virus going to explode and infect and kill off half of the world?
No.
Thank you.
Excellent.
Very good. Why don't you think polio can be eradicated?
Well, I think the biggest problem, of course, is always changing governments and changing policies in different countries.
Yeah.
And polio is a very important example that I usually use when we talk about countermeasure development for these BSF-4 agents. I'm more of a fan of therapeutic development than I'm of vaccine development, for that very reason that with polio, the doses they don't cost much theoretically for an individual shot somewhere, but it's very difficult to get the vaccine to all the people that really need it. Yeah. That's especially true, of course, in Africa, where people live in villages that can only be reached by boat or that are completely secluded. So to have full coverage is difficult, even with something that is incredibly cheap and we can produce very easily. And then, of course, on top of that comes the cultural problem that many people don't want to get injected with something they don't understand. There's beliefs in certain parts of Africa that you steal life force with injections, so they will think double and triple before they let you give your shot. Yeah. And so I think developing a vaccine for something like Ebola, where you spend multi-million-dollar programs to have something where at the end a dose costs I don't know how many dollars, is a little bit unrealistic in terms of controlling this virus. But it's, of course, my own personal opinion, as everything else I say, by the way, here.
Nothing is not that of your employers, yes.
That's right. Nothing is Tunel or NIH opinion here, what I'm saying.
So, but aren't Ebola vaccines being developed somewhere?
Yes, of course. And so there's we develop them, of course. We help developing any countermeasure. And, of course, it would be fantastic if we had one. It's always nice to have a vaccine. And there are several needs for that. The first obvious need, of course, is for laboratory workers. You want to be safe, ideally, and ideally have a post-exposure prophylaxis and these things ready. The second thing is that bioterrorism, bioweapons angle. That what do you do if somebody actually attacks with such a thing? Can you do ring vaccinations to contain it? Or, of course, if you're aware that the threat is coming from somewhere, can you prophylactically vaccinate the population? So these are certainly games that people play in their heads, like they're kind of war games of what if. And yeah, of course, if we find out that these viruses are way more prevalent in African countries than we right now know, then maybe it is worthwhile having these. That said, I think still antivirals would be better because they're easy to produce, they might be cheaper, and it might be easier to convince people to take them.
And you may be able to, even if you can't convince the average person to get the vaccine, you might be able to convince medical personnel, which could help halt an outbreak in the case of Ebola.
Correct. That's right. So it's always first responders. It's similar to like what we do in the U.S. with the smallpox vaccinations, which first responders get in the case. And of course, those people are highly educated medically, and so therefore they are much more willing to take these vaccines.
So a related question is from Melissa, who actually sent this a couple of weeks ago. I sent it to you, Jens, but I saved it for today. Who writes, "Good afternoon. I've started listening to your podcast and have a very layman's level of understanding of viruses in the immune system. Given the recent Ebola outbreak in Africa, I'm wondering why they can't use the Reston Ebola virus to make a vaccine of some sort, given that this strain doesn't make us sick.
Yeah, it's a good question. So, for background, I guess, so Reston virus is a virus that was discovered in the U.S. after it was imported from the Philippines, and after that, it got imported into Italy as well. So there are a few outbreaks of this among non-human primates, and there are no human infections known that have definitely been documented in a peer-reviewed manner. There are 3 or 4 different cases, mentionings, or anecdotes of seroconversion of people that have been dealing with these outbreaks. And there was a large Reston episodic in the Philippines a few years ago among pigs in the Philippines as well. And because this was such a large episodic and so many pigs got infected with this and so many people were involved in culling them and none of these people died, there's a general feeling that Reston is not virulent for humans, not pathogenic for humans, or not as virulent as the other viruses. That said, there's no real proof of somebody getting an actual, dose of that virus and then living happily ever after. So my first response to this is I wouldn't want to get Reston virus injected into me in any circumstances. So it can be, maybe it was just luck that nobody got infected in some kind of a way, and we don't know what it does. So that's I think the first part of the question. The second part is I'm not really sure how solid the data are that we can get cross-protection between different viruses. So there are different studies out there. To my knowledge, so far we have never accomplished with a monovalent vaccine cross-protection between Marburg and Ebola viruses. So then you're bound to only that one group. So maybe your Reston injection protects you against Ebola, but then you might die of Marburg. And then within the group of the Ebola viruses, some vaccine candidates have shown limited cross-protection between Ebola and Sudan, but I'm not aware of one that with one GP, for example, protects against all the different Ebola viruses, 100% in non-human primates. And so, of course, that might be the case with Reston. And then the third problem with this is, how are you going to test it?
Yeah.
Yeah. I mean,
this is, of course, the biggest problem with vaccine development or therapeutic development for all of these viruses. Yeah. Because you cannot do human clinical trials, obviously, because that would be clearly unethical and people would die. And at the same time, then you're reliant on non-human primate models or other animal models in the lab, and you try to find decent biomarkers to extrapolate your results to humans in the case. But if you do that, Reston is a pretty efficient killer in non-human primate models. So
that's a general problem for any Ebola or filovirus vaccine, is that even if you make a virus-like protein or a purified protein, how do you test it? Because the number of cases are so few, right?
Right. You can't do a typical field vaccine trial either and just vaccinate susceptible populations and compare rates, because it could be years.
Right. Because if you don't know the reservoir, you also don't know when it could possibly pop up and where. Yeah. And so the outbreak in Guinea certainly, I think, caught everybody by surprise. I told you that there have been niche studies predicting that these viruses might be around, but these studies were 10 years old and we haven't seen anything. So I don't think there was anybody out there who said, oh yeah, Guinea, obvious.
Right, and we don't even really know what the natural incidence rate is. So you couldn't necessarily compare. I think that's kind of what you just said. You couldn't necessarily. What are you comparing to?
I wanted to ask you, in any of the bat metagenomic studies, viral metagenomic studies that have been done, have it picked up vilo-like viruses outside of Africa?
Not to my knowledge, but I wouldn't be surprised if it's coming. So, most of the people who are doing these metagenomic studies, they have a particular virus group in mind that they're targeting for.
Sure.
So, if you look at Christian Drosten and his fantastic paper on paramyxoviruses, he focused on those. Many people right now focus in bats on coronaviruses. So, I'm sure they're going to look at these samples also for other viruses eventually. So, I wouldn't necessarily be surprised if somebody like Christian Drosten would come up with a big paper and say, we have found 20 new filoviruses.
Right.
On the other hand, I would also not be surprised if it's the opposite and they don't find this because it's a rare reservoir and we simply don't know where this hides.
Yeah. So you mentioned at the top that you work on other viruses that no one has ever heard of.
Yeah, I work on a few. Well, the BSL-4 viruses in general are not very well known. I mean, there are a few that people have heard of. Ebola, of course, Lassa fever. But I guess if you look at other viruses like Omsk hemorrhagic fever virus, or Kaisano-Forest disease virus, or SFTSV, there are a lot of these, I call them niche viruses. They are exotic, they pop up somewhere, or they are very localized to a particular region and haven't really grabbed the attention of public health officials. And so
SFTSV is the one in China and also in the Midwest of the U.S., right?
Yeah, the one in the Midwest of the U.S. is different. That's heartland virus.
So it's a different virus.
Yeah, it's a different virus, but it's closely related. So this is a group of flevoviruses that is expanding rapidly now that people look more into flevoviruses. So there has the Bunya viruses in general. So this is the family is the Bunyaviridae, and there are 5 different genera in that family: the hantaviruses, flevoviruses, nairoviruses, the orthobunyas, and the taspoviruses. And it's a very large family because in the 60s, 70s, and 80s, a lot of people, classical arbovirologists, have done a lot of isolation studies, and there was funding for ecological studies at that time. And so many of these viruses in this group might actually be pathogenic or might cause disease, but they have just been characterized so far to say this is a new Bunya virus, it belongs into this group, that it was published in the International Arbovirus Catalog, and then they went on to the next virus. And so now it turns out that a lot of the new emerging viruses that fit into this family are actually closely related, or are some of these viruses that we theoretically know about for like decades, but nobody has really looked into them. So SFTSV falls into a group of a virus that there's about 100 papers on there called Banja, that causes febrile disease in people, and it's around in Europe and other places. And it was a very obscure group, and now Heartland virus belongs to this, which caused a few cases in, I think it was California or something, somewhere in the U.S.
Missouri, Midwest.
Yeah, so that virus, then SFTSV, which appeared in China and causes actually a lot of cases in China. Unfortunately, most of the literature on this virus is published in Chinese in journals that you don't see on PubMed. And then now a few Russian papers appeared with a whole bunch of other viruses that belong into this little subclade. And so these groups get bigger and bigger. And I think we will see this with all the metagenomic sequencing. We will see this very soon, that all our clades and families and genera of viruses that are dear to our heart will suddenly explode and will have to be divided and have to change and so on.
Yeah, so I didn't realize that so many papers were published in other languages still. I assumed, I guess naively, that it's all in English. How do you find these?
No. Well, so the first thing you can do, of course, is look at the reference lists of the papers that you are finding in PubMed.
Yeah.
And so if you read an SFTSV paper, or if you read a paper on Crimean Congo, the likelihood that you will find papers in Turkish journals, in Crimean Congo, for example, Turkish journals, or even Iranian journals is relatively high if you look at epidemiology and clinics and so on. So you can find that. The second thing you can do is if you work at an institution that is very well equipped and has some finances, like yours, you can go away from PubMed and you can start looking at Web of Science and all these other databases that are at cost. And they cover many other journals that are not covered in PubMed. And then, of course, you can go into the individual databases of the national libraries of individual countries, and you can start looking there for your keywords, depending on whether you speak the alphabet, know the alphabet, or speak the language, or use Google Translate in a cunning way or so. So there's a lot of data out there, and this is simply dependent on it's dependent on two things. First, of course, is national pride and requirements for careers in a particular country. So, for example, when I worked in Russia, that was in 2001. At that time, it was still necessary for Russians to publish in Russian journals if they wanted to become academicians. So that causes, of course, a conundrum. On the one hand, you need to publish in Russian to become that. On the other hand, if you don't publish internationally, then nobody knows you and you don't get involved. And the big problem, of course, is you're not allowed to publish dually because all the journals prohibit this. So you have to pretty much take one of the two, and you will lose with that. And so that's an example for Russia. I wouldn't be surprised if this is similar in many other countries. And then, of course, national pride. There's this, with France, they had a big initiative, like I think 10 years ago, to have all their conferences back in French and to boost the national language. So, of course, there are very important French journals on epidemiology. Most of them are covered in PubMed, but there are others that are not. So that's one of the things. And then, so the other problem is that you might want to publish in a journal that is in a different language because you reach a particular peer group.
Yeah.
So if you want to publish an article on clinical manifestations of Crimean Congo hemorrhagic fever in Russia, it might be smart to publish in Russian so the local clinician can actually understand this.
Right.
That's amazing. Well, I have my hands full with the English literature, so.
Yeah.
But the thought that you're missing something is, kind of disturbing.
A lot of people would argue that most of the scientific literature is not written in English.
Since I edit all day, I would agree with that. But this is an important thing. So if you look at blatant self-promotion here with my filovirus book, but there's a CD in that book that has the list of all the references on filoviruses until 2008, and there were about, I would say, 4,200 or so references in there. And of those 4,200, you would see only about 3,000 or so in PubMed. So every fourth you would not see. Now, I listed everything from abstracts to whatnot. So, this is a little bit skewed. But there are many important papers, especially in Russian, on filoviruses that people do not know about. And these are pathological studies, necropsies and so on.
Yep,
and if you look at Crimean Congo, I would say it's probably half-half what you find in PubMed and what you find somewhere else. Mainly because of, first of all, the increase of CCHF cases in Turkey and a vast literature in the Turkish language that is developing. You can find all of those articles, 90% of those articles, freely on the web though. So there's something called Turkish Medline. So you can go there and you will find them, and they have PDFs to download and you can find them, but they're not indexed in PubMed. And there's a similar system in Iran. So you can find a lot of CCHF articles in Farsi that you can download in the archive. But it's, of course, the problem with all of these articles is you have to learn how to differentiate the good from the bad.
Of course.
That's already difficult in the English language.
Yeah,
right.
Yeah, right.
So if you don't speak it, then it's hard if you don't have direct contacts with other people. But it is a warning about these exotic viruses, that a lot of articles that are really, really important is something that you do not see on PubMed.
Okay.
I want to get back to you, but Alan has to leave, so we'll ask
Yes,
him.
I have an errand to run, so I will go ahead and cut out.
Okay, you want to give your pick?
Yeah, I'll go ahead and do my pick. This one, I've picked the American Museum of Natural History before. That's the big Natural History Museum in New York, but they've got this thing on their website now. It's their digital special collections from their library. And like a lot of institutions like them, they are now going through their vast archive of images and scanning all this stuff. And you can now browse through these massive collections of really cool stuff. It's everything from sketches and drawings done by an African expedition to pictures of people painting the murals that are behind the dioramas that you see in the halls. It's I could just get lost in a site like this.
Yeah,
It's
there's
cool.
neat lantern slides with
Yes.
all the original numbering and cataloging information, and
Yeah.
it's gorgeous.
Nice.
Yeah, really neat.
Nice. Look at this. Italian woman, Ellis Island. Amazing. Wow.
Yeah.
That's cool. Thank you, Alan.
Sure.
Alan Dove is at turbidplaque.com and also on Twitter. Thank you, Alan, for joining us today.
Always a pleasure.
Good luck with your errand.
Yep. Thank you.
All right. See you next week.
And
Bye.
good to meet you, Jens.
Good meeting you.
Take care.
Bye, Alan.
Bye.
Bye.
So, Jens, can you give us a sense of what kinds of experiments you do? I don't know. I know you might not want to talk about it, but is there something you can tell us that would give us a feeling?
Well, there's several things I do. So you asked me about, the other viruses that we're playing with. So I have a small program on simian hemorrhagic fever virus, which is an arteriovirus, so a nidovirus, like coronaviruses. And we try to establish that as a hemorrhagic fever virus model in animals and also, of course, in vitro, because this virus does not infect humans, and so it's a safe thing to do, and it's easier to play with, of course. This is kind of feeding into the overall idea that has been about has been around for quite some time, that there might be a common denominator among viral hemorrhagic fevers. I'm not really sure whether that is true or not, but it's possible. And so if that is true, then we might find it with that uncomplicated virus. And then I told you I'm a molecular biologist, and so I got all my training in Mike Farsen's lab, so I'm a virus entry person. So we're looking for receptors, of course, of many of these high consequence pathogens and restriction factors in cells, and things that Wilkin would like, just not with HIV.
So to look for a filovirus receptor, you would use pseudotypes where you have just a glycoprotein present?
Yeah, so most of my experiments are reliant on pseudotypes, not only with filoviruses, but many other things. I find it a very nice, easy system to play with, and it's a very good control system because, of course, you change only the glycoprotein on the surface of your particles, and you can screen several viruses at the same time and then determine at least tissue tropism and where's the receptor present, and then you can, if you have an antibody against different things, you can try to do blocking experiments and so on. And eventually you do the dreaded IP and then don't pull anything down and get all disappointed.
Right.
So this is something that we definitely work with. And then a little bit removed from that, you can do VLPs and infectious VLPs. Infectious VLPs is a little bit misleading because, of course, they're not infectious, but things that you can pass from cell to cell. And then IPs and virus overlay binding protein assays and these kind of things. It's tricky. I mean, I've become very humble when it comes to receptor search. Mike, of course, discovered the SARS receptor at the time, and then my girlfriend in his lab discovered the Machupo virus receptor. And so these were big things. And so I think we got a little bit excited and a little bit too cocky about how relatively easy it could be to find receptors. And then we learned that it's not. So,
the easy ones are already done.
Well, I'm not really sure whether they necessarily were the easy ones. It's just many of these things are, there's a mixture of choosing the right protein, choosing the right virus, and having the right technology at hand at the time. I mean, maybe there are other easy ones out there. We just don't know which ones the easy are. So with which virus do you start? And the Machupo virus story is a very good example. With Machupo, we were able to, my girlfriend was able to identify a receptor binding domain that was nicely expressible and could be used as bait in a co-IP. But later on, we tried to do the same experiments with the other arenavirus glycoproteins, and it turns out that in none of them could we identify something that was remotely as expressible, if expressible at all, or bound to the receptor. So, it was simply choosing the right virus of those 7 virulent ones, and otherwise we would have played with it probably for a few weeks, and then Mike would have said, "Oh, this is a waste of time," and it would have been over.
Yeah.
So I think it's always a mixture of ability and luck, and so I'm still hoping to get that luck one of these days.
Keep at it, you'll get lucky.
Yeah, that's right.
I just got an email from Matt Freeman who says the CDC has announced the first case of MERS coronavirus infection in the U.S. Imported from a traveler from Saudi Arabia in Indiana. The head of CDC said, "We expected this and we are ready." I think
that's true. I think that's true. I think they're ready.
Yeah, I mean, it's going to happen. If you have enough cases, it's going to happen because people travel. What I'm amazed is that it hasn't happened all that much with H5N1 influenza, just that case in Canada last year, I guess, right?
Yeah, but again, it's a question of surveillance and how sick people really get when they get it. Right?
Yeah.
I mean, they could be here, they could have been infected, and they felt like it was a bad cold and bad flu, and they stayed at home for a few days, and who knows?
This MERS case isn't going to go anywhere, I'm sure. It's just there's something about, I think it's like Ebola. You get that primary zoonosis, and then
but it will be a big headline.
Yeah. So Matt says, does this make it into this week's TWiV?
Yes, it did.
It just did.
And then he said, "Still think it's not going to spread?" I said, "Yeah, I don't think it's going to spread. I think it's going to travel, and you'll get these cases here and there, but it's not going to spread." Matt would like it to because he works on coronaviruses. Right? It's not nice.
I don't think he would like it, but yes. I mean, of course, it's every researcher looks at their viruses and kind of hopes, of course, to get funding for things. And so, in the end, it is an important question of when as you brought up HIV before with the 4 introductions. I mean, you never really can predict whether a virus will manifest itself and will suddenly spread faster. And I think a few weeks ago, all of us would have said, well, this MERS thing is trickling out and it's going to be completely gone. And now we realize, wait a second, now we find more cases. So maybe we're going to have more cases of it. But I agree with you. I don't think this will be any kind of exponential spread and suddenly we'll deal with a big MERS epidemic in the United States. At the same time, of course, if cases come over here and stuff, I would like to see them isolated.
How do you think I feel with polio being eradicated or close to? Here you just said, most investigators like their virus to become important, but mine's going the other way. Well, I guess that's good because I'm an old guy and I have to retire at some point.
Well, but you could also look at the other way. Like you could find the conditions on how you can still work with your virus, and then you're very special because you're one of the people in the world who's still allowed to work with it.
Yeah, right. All right. Kathy, anything else from you?
I think that's about it.
I have 2 email here, so I'm going to just do these 3 Ebola related. So we did Melissa's, then Charlie. Kathy, why don't you read Charlie's email? It's the first one.
I lost my Google Doc. Oh, there it is. Sorry. Google Doc. Charlie. Sorry. Delay here.
No problem.
Dear Mr. Racaniello, thanks for the great work you and Mr. Despommier do on TWiV and TWiP. I'm an engineer by training and have always been an amateur scientist. My primary interest is insects, particularly myrmecology. Oh, too bad Alan's not still here. Myrmecology, what's that?
No idea.
It sounds like ants or something.
Studying parasites of insects led me to TWiP. It is by far my favorite podcast. Yeah,
it's ants. Good. You're right, it's ants.
Yeah. TWiP led me to TWiV, which is also fascinating. In light of the current Ebola outbreak, I would be very interested to hear your thoughts on the big picture of the probability of a dangerous global viral outbreak. I think we already heard that.
Yep.
Yeah, we covered that. Not.
I realize we are much more mobile than we were in, for example, the 1918 Spanish flu outbreak, but also our healthcare tools and systems are more responsive. It is very hard for someone as ignorant as me to speculate as to where the viral arms race now stands. Regardless whether you choose to address that or not, I want you and Mr. Despommier to know that your podcast work is very much appreciated. I know it takes effort to put together and record the podcast, and you have many choices about where to apply your time and talents. Thank you. From Charlie.
That was nice.
Yeah. Mm-hmm. Then John, our friend from Kansas, sent us a story. I guess it's an AP story. Crowd attacks Ebola treatment center in Guinea. Angry crowd about an Ebola outbreak has killed 86 people. Well, this is quite old. Attacked a center where victims were being held in isolation. The mob accused Doctors Without Borders, health workers, of bringing Ebola to Guinea, which, of course, Jens, we know is not true. It's not possible, right?
Right. But this is a big problem because we talked briefly about colonial fears and so on. Yeah. And this is something that happens in most of these outbreaks, not only filoviruses, but also other dangerous viruses that are not showing up very often. Of course, conspiracy theories are always abound. And we have this here, we have this in the United States where vaccines allegedly cause autism, and people believe this by tens of thousands. People believe this, and so the same way you have these kind of rumors in Africa, where whenever something bad happens, it must come from the evil West, and we again once oppressed, and so there have been incidences during outbreak control where I'm not really sure whether this was Doctors Without Borders, might have been WHO, got shot at when they tried to land and tried to contain the outbreak. There's also a lot of rumors. There are a lot of rumors going on in different countries about organ trade. So if we go back 10 years, what people did is, especially Doctors Without Borders and other relief organizations, they built isolation tents.
Yeah.
And of course, they brought a living person with an ongoing Ebola infection and with clinical signs into the tent. And then 2 days later, they brought a dead person out of the tent. And of course, nobody knew what was going on in the tent. You look outside, and so if you are inclined to believe that the people who came here are in there for something bad, then they did kill that person in that tent. Mm-hmm. So this was dealt with very elegantly and very swiftly by simply putting windows into the tent, and all of these things disappeared because then relatives could look inside of what is happening and see that their loved ones are being treated and not molested. And so I think there were a lot of lessons learned during all of these individual outbreaks, and every single new outbreak, of course, depending on which country it is, brings new cultural challenges and new rumors and new ways of alleviating this. And Doctors Without Borders is doing a fantastic job in beginning to educate local population in the traditional languages in the particular area, using songs and radio broadcasts. They are not going directly into the village anymore and teach people, but they go through the local physicians that have the trust of the population, and so that helps a lot. But of course, you cannot avoid all of the conspiracy fears that people have.
Okay, let's wrap this up with some picks. What do you have for us today, Kathy?
Well, I have the creative student presentations from my class, or just a little discussion about that. So we gave students 4 different options for some of their final points in the course, and we invited some of them to do creative presentations, and it was in part based on the Singing Microbiologist link that someone sent in to TWiV a while back. And so I was inspired to write some words about HPV to Yankee Doodle, and that's, I think, what we can somehow try to link to for people. And I recorded it to a karaoke soundtrack with my brother so that I could have someone else singing with me in the class and be brave. And the class sang along, and they really enjoyed it. And so maybe I'll just recite a couple of the chorus lines. It's not opening up here. There it is. The first verse: papilloma causes warts. It also causes cancer. Should you get HPV vaccine, I can give you the answer. And then this is a Yankee Doodle. Keep it up. So papilloma goes with sex, infecting men and women. And though it has no envelope, you won't catch it when swimming. So the reference is, of course, to polio, but it's which you could catch swimming. So, and then it goes on, talks about the DNA and the differentiation of basal cells and E6 and E7 and so forth. And so I didn't get that out in time to inspire the students, but we had 4 student groups take us up on the presentation thing. And one was a student who put together a bunch of video based on some clips from a TV show and stuff, where he was pitching an ad campaign to the CDC about H7N9 influenza. And we had a certain set of things that we wanted them to include in, wasn't necessarily quite so required in the creative presentations, but they did that. Another person put together a game, kind of a chutes and ladders game, about the consequences of behavior and knowledge about HIV. So, and there were different character cards that you could draw, somebody with promiscuous behavior or somebody conservative and so forth. There was a person who did a long epic poem about a MERS researcher falling in love with a SARS researcher. And it was really well done, and he had a PowerPoint to go along with it, and so he brought in all these facts, and I wrote down that he rhymed virus with desirous, and then one line was something about, we work with SARS. It seems your virus is similar to ours. But my favorite of all was a pair of students who did a skit about poliovirus, and the other professor, Beth Moore, and I would constantly be saying in class, oh, well, that's anthropomorphizing the virus, but we're just going to do that. And so they started out by saying, we're going to anthropomorphize like crazy. And so it was poliovirus going in to talk to its psychiatrist and saying things like, do you know what it's like? I used to be in all the countries, and now I'm just in the developing countries. And internet there is terrible. And I feel really welcome in the cell because there's a molecule that's named after my receptor, the poliovirus receptor. Things about, I'm unlike flu, I'm faithful with just one host. The virus, I can travel really light because I don't have to bring in my RDRP. And then he was saying, like, oh, but as a kid, on the playground, I was so insecure, because my messenger RNAs weren't capped and all the other kids had caps, and then he said, do you know what it's like just having an iris? I mean, having an iris just come having a ribosome come and hit you right in your iris. Ouch. And it was just, I thought, really good at getting, some facts across that maybe would help students to remember it. So I was a little disappointed. A couple students had an option to and they chose it to just take the final. And we put some more questions about polio, and 2 of them still missed it and thought that poliovirus is capped. The RNA is capped. So
it's always going to happen. Yep.
Even with this great mnemonic. But I think it was a success, and we already did get a little bit of feedback from the students. Did they like the 50-point option? Another option was they could listen to 2 TWiVs and write a write-up on one of the papers discussed on that TWiV and propose some future experiments.
Oh.
So those were pretty well done. But we asked students, did they like this flexibility option for the final points? And they did. And one of them said, but could we make everybody have to do a creative presentation?
Yeah.
Those are the best.
Yeah. That's pretty cool.
So
we
had a lot of fun with it.
Are you going to put the lyrics somewhere, or do you want us to?
I put them in the Google Doc, but then it looks like I have to share that with people. So if you can figure out a way to post the lyrics somehow.
All right, I sent you a request so I can sort that out. Okay, cool. Thank you. My pick is a book I came across this past week while doing some research. It is an entire book on virus entry into host cells. It's relatively recent, 2013. It has chapters on attachment, picorna entry, reovirus, influenza, filovirus, paramyxo, retro, and it's open access.
Yeah.
Everyone can get this.
Yeah.
I'm amazed. It's Springer. And usually they charge a lot of money for their books. But
I'm a big Springer fan. I publish with Springer, so go Springer.
Yeah, your book I was just looking up. I found it on Springer.
Oh, don't say the price.
I won't say the price. But it's not. I hoped it was open access, but it's not. But this one is. So if you're interested in viral entry, check this out because you can download the whole thing. Everybody, apparently. So check that out. Jens, you have anything you want to share?
You catch me off guard.
You could pick your own book if you want.
My own book? No, I can't do that. It's a little bit too too self-indulgent and stuff. So, well, I really like. I don't know. You probably covered it, but you had Eugene Kuhn on the show a few weeks ago, right? So you must have advertised his book, and that's great. So
yeah, we could do that. Eugene Kuhn's it's a book on evolution, right?
Yeah, I found this very, very interesting and a lot of fun to read, especially his thoughts about how viruses came about in the end.
Yeah, that's almost as expensive as yours.
I'm sorry.
But that's okay. It's not your fault. That's a great book. I really like that. Yeah. All right, we have a listener pick from Joseph. Dear Titans of TWiV, a listener pick. He sends a link to a paper. Mutations explain poor showing of 2012 flu vaccine. The notion that mutations allowing efficient propagation in eggs, or for that matter in cell culture, might be incompatible with full vaccine efficacy has been a theoretical concern for a long time, but this is the first demonstration of the problem in the field of which I am aware. It's very possible that I am not fully informed, but one more good reason among many to consider alternative vaccine technologies. This is a very interesting story of how the H3N2 vaccine was matched very well with the circulating virus in 2012, but propagating it in eggs selected for changes that altered its antigenicity, apparently. And so, yes, flu vaccine technology is ancient. We need to really change that. So it's a pretty good story there. Thank you, Joe. And Kathy, you had a pick also from
another listener, Phil, actually here from U of M, sent me an NIHOD video, and I think NIHOD is just the NIH's YouTube channel. It's called Emerging Disease or Emerging Diagnosis. It's a public outreach video. I think it's about 3 minutes long. And they talk about Ebola and Lassa, and it's for the general public, basically describing or discussing, were these viruses always around or have they recently just emerged? And they kind of come to the conclusion that, that Lassa is maybe 1,000 years old and Ebola is maybe 10,000 years old based on molecular clock calculations.
I would differ with the Ebola. If the integrated filovirus genes that we find in mammalian genomes are an indication that they
That's true,
You know, depending on how closely you can say that something is related to something that we know today. But if that is halfway true, then these viruses are very, very old.
Okay. So, and it was put together, there's credit at the end by Stephen Gear in conjunction with the Sabetti Lab at Harvard and the NIH Common Fund. So it's, I think, worth sharing with your non-science friends. So,
yeah, that's apparently the NIH YouTube channel. NIHOD, cool. It's good that they have one. All right, that will do it for TWiV 283. You'll find this and all the other episodes at twiv.tv and also on iTunes. And do send us your questions and comments to twiv@twiv.tv. Our guest today, Jens Kuhn, is the principal at Tunnel Government Services and also lead virologist at the Integrated Research Facility at Fort Detrick. Thanks so much for joining us, Jens.
It was an absolute pleasure.
I really enjoyed it. I learned a lot. And come back sometime.
Sure.
Now that you have approval from the government to use Skype, right?
Well, yes, but I will have to get separate approval.
Well, you have to do it each time, but it wasn't so painful, right? Anyway, thanks very much. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was fun.
And I am Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.